a little bit here. And so we were talking about the ways that the body regulates the uterine cycle. So remember that we have the proliferative phase where when estrogen levels are high, the lining, the endometrium gets thick. And then we have the secretory phase where it gets vascular and glandular. So it starts to secrete glycogen, and that will feed the early embryo prior to implantation. And then we can have menstruation. But if we don't have menstruation, if we actually get the implantation of an embryo, then we can maintain pregnancy. And so the maintenance of pregnancy is due to the embryo producing a certain hormone, and some of you might have read about this hormone because it was part of your take-home quiz, which is called HCG. And so this stands for human chorionic gonadotropin. And this is produced by the embryo. And if the embryo cannot produce enough of this hormone, then it will, the embryo will be aborted. And so they think about 33% of all embryos produced and that make it this far into the uterus and get ready to implant are spontaneously aborted. And so they make reference to this in the video that we watched last week, which talked about how sometimes the embryo doesn't implant, or if it implants and doesn't produce enough HCG, it's got to produce enough HCG in order for the lining to be maintained, then if the lining will just slip off and the embryo will go with it. And so they think a lot of embryos are produced that are not viable enough, that are not healthy enough. And so we're going to talk a little bit about that because we're going to review meiosis, and we're going to review oogenesis and spermatogenesis, and then talk about some of the mistakes that can be made. Okay, so when we talk about this, we need to talk about how HCG does this. And so what it does is the HCG maintains the corpus luteum. And does anybody remember what two female hormones are produced by the corpus luteum? Progesterone and estrogen. So estrogen and progesterone levels stay high. So if they fall down below a certain threshold, then the endometrium will be slept off during menstruation. So prior to menstruation, estrogen and progesterone levels kind of collapse, right? So that's what kind of gives you the PMS, the premenstrual syndrome, where you get really irritable because your estrogen levels are really low. Okay, so if the estrogen and progesterone levels stay high, this maintains the lining of the uterus. So this is actually thought to be a really important screening of embryos, because the embryo in essence actually has to prove itself, right? So it doesn't just get automatically, embedded into the uterus and then just automatically nourished and placenta formed. But the embryo actually has to go through this proving itself to produce enough of this HCG, and HCG levels continue to rise during those first 3 months of pregnancy. And then after the placenta is formed, so after that first trimester, we'll put first trimester, when placenta forms, HCG levels decrease. So they're only high initially, right? And the placenta then produces estrogen and progesterone. So the placenta takes over the role. Takes over the production of progesterone and estrogen. And so they believe that this HCG, and specifically the high levels, also probably have something to do with being kind of ill and sick during that first part of the pregnancy. So you have a lot of morning sickness where you vomit, right? And you also can't eat a lot of the foods that you would normally eat. And that is perhaps a mechanism to keep females from eating or drinking things that could possibly harm the early embryo, because the early embryo, like the nervous system, is developing really early on, and it's really important that environmental toxins don't get in there. And so like females might not be able to, if they're early on during the pregnancy, they might not like to drink coffee, or they might not like to eat certain vegetables, or they might not like to eat spicy foods, or they might not like to eat or drink alcohol, right? So that would protect the developing embryo from those possible things that could lead to developmental problems, okay, and possible birth defects. Okay. So it has to do with this HCG. And you might have heard of HCG previously. I don't know if it's still popular, but it was popular a few years ago as a weight loss mechanism. And so both males and females were injecting. They would like get in syringes full of this, and they could give themselves injections of it, or they could take it orally. And it would make them lose a lot of weight really fast, because what it does is it stimulates early pregnancy. So it kind of boosts up metabolism, and it decreases their appetite, so you actually don't feel like eating. Because, you know, sometimes when females are early on in pregnancy, they'll actually lose weight because they don't eat, because they're not hungry because of these HCG. So HCG in males and females was used as a diet aid, right? So a hormonal diet aid.
Do they not do it anymore?
I don't know. I remember, you know, I know people that did it in the past, but I don't know if they still do it. If it's what happened with the people that I know is they, after going off of it in like 6 months, they just gained all the way back, right? So it wasn't like a long-term way of losing weight for very long. Okay. The other thing that's interesting about this is that this could be an example of what we call a maternal screen, and I mentioned this just briefly. A maternal screen of embryos, so that only viable embryos are able to survive. One of the interesting things about this, however, is that there is one genetic defect that seems to get past this maternal screen, specifically as females age. And so that would be a Down syndrome embryo. So when we look at Down syndrome, okay, this in older females, particularly over the age of 40, can produce enough HCG to be maintained. So this is where the embryo would have an extra copy of the 21st chromosome, and we're going to talk a little bit about how they are able to sample fetal cells and get chromosomes for maternal testing. And so the idea here is that perhaps as females age, their screening mechanism becomes less intense, because if you think about it, their probability of getting pregnant again goes down, right? So if you're over the age of 40, or if you're approaching 40, then the likelihood that you're going to be able to get pregnant again goes down. And so maybe you're not going to be screening as thoroughly as, say, a 20-year-old female would be doing. So that's another hypothesis. That's one hypothesis for why there are more babies that are born that have Down syndrome by older females. The other hypothesis is that as the eggs, and we'll talk about this, as they stay in the ovary, perhaps they are more likely to have genetic abnormalities during that final meiotic stage, that they undergo right after fertilization. And so maybe they're just, as they get older, the eggs also get older, and they are more prone to having genetic abnormalities. Okay, so kind of two different hypotheses for why Down syndrome is more common in older females than younger females. Okay, any questions about that idea?
Is it because it can produce enough or can't produce enough?
It can produce enough
okay.
HCG, yeah.
Okay.
So it has to, in order to be maintained, it has to produce high levels of it. But they're thinking that maybe it doesn't produce high enough. If you're 20 and you have a Down syndrome embryo, it wouldn't be able to get past the screen and it would be spontaneously aborted. Okay. But if you're 40, maybe the screen is lessened so that you're more likely to maintain the embryo. Yeah. Okay, so let's talk about the human life cycle. So this should be a review for most of you who have taken cell biology. Hopefully most of you have. And so we have in the adult stage, I was hoping this would be a better pen. So the adults produce gametes. And these gametes would be the egg and the sperm. In humans, these gametes have 23 total number of chromosomes. It is different for different species. Chimpanzees actually have 24 chromosomes, and some other organisms, like some invertebrates, have many, many more total chromosomes than we do. But so that tends to be species specific. So there's 23 total chromosomes in the gametes. The egg and the sperm then come together to produce what is called a zygote. And the zygote, unlike the gametes, has 23 pairs of chromosomes. So you get one copy of each chromosome from one parent, right? Which is oftentimes why in your textbook you'll see that there's a red copy from the mother and a blue copy from the father. So that's why they're colored in the way that they are. Okay, so these cells are said to be haploid because they have half the genetic material as the zygote, which is said to be diploid. Now, to go from the adults that are producing, in order to produce the gametes, there's a special type of cell reproduction, and this is called meiosis. So this is the type of cell reproduction that occurs in the seminiferous tubules of the testes and also occurs in the ovaries. So meiosis is only the only type of cell division that produces haploid cells. Then the zygote is going to start to undergo cell division, and we're going to talk about this early embryonic development when we get to the next chapter. But this development from the fetus into the newborn and then back into the adult, all of this is accomplished by a different type of cell division, which is called mitosis. So mitosis is really important because it allows for organisms to grow, to develop. And then also, even as adults, we have mitosis occurring, like, for example, in the bone marrow. The red blood cells do not divide because they have lost their nucleus. And so there are hematopoietic stem cells in your bone marrow that undergo mitosis to produce new red blood cells and new white blood cells. Likewise, our skin is constantly undergoing death, and we shed it, and then we replace the skin. And so lots of our tissues, even the bone, undergoes, the cells die and then are replaced via mitosis. Okay. So this is all mitosis versus that which would be the meiosis aspect. Okay. So we're just going to overview the difference between the development of the egg and the sperm during meiosis. So the things you need to know about meiosis is that there are 2 consecutive cell divisions. Okay, so we have to start out with a diploid cell, and this diploid cell is going to undergo 1 cell division to produce 2 cells that are slightly smaller, and then it is going to undergo a 2nd cell division to possibly produce 4 cells. So these would be the haploid cells. Right? And this first cell division is what is called meiosis 1. Meiosis 1. And the second cell division is called meiosis 2. And this will be important because when we start to talk about how the cells originate in the ovary, for example, all the cells, when you are born as a female, all of the cells are arrested in the early stages of meiosis 1. And then during a monthly development by the production of FSH, as the oocyte develops from a primary follicle to a secondary follicle, and then we get the release, meiosis 2 actually doesn't complete until after fertilization of the oocyte by the sperm. Okay? So if you remember that each of these stages, each of these has a prophase, which is an early stage of meiosis, so that would be like prophase 1, and then metaphase 1, and then anaphase 1, and then telophase 1. So telophase would be at the very end, where we get, telophase 1, where we get the formation of 2 cells from 1 diploid cell. Okay, and likewise, we have the exact same here, but it would be instead of 1, it would be 2. So it'd be like prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay. So this is theoretically what can happen during meiosis, but it's different if you're looking at the formation of sperm versus the formation of eggs. So we're going to talk about now what is called spermatogenesis. Let's get through this real quick. Okay, so let's talk about spermatogenesis. So this takes place in the seminiferous tubules. And it takes place inside of a cell that lines the seminiferous tubules. And these are what are called the sustentacular cells. These have another name, which are called Sertoli cells. Okay. So spermatogenesis, the formation of the sperm, actually takes place in another cell. And this is important because this sustains it. So think about, oh, what does it do? It sustains it. It provides nutrients for the developing sperm. And it also actually, interestingly, protects it from the immune system, because the immune system could potentially produce white blood cells that would attack sperm as being foreign. And so there's a kind of a blood-testes barrier, like a blood-brain barrier. So the sustentacular cell keeps white blood cells from attacking the developing spermatocytes. Okay. So if we look at a diagram of this, let me show you the sustentacular cells first. Oh, I don't have a picture of it. I did. Okay. So in your book and in the diagram that I gave you in lab last week or the week before, you had this all was occurring inside of a cell. So when we look at the diploid cell, the diploid cells are called the spermatogonia, or singular is the spermatogonium. Okay, So this gonium, this is diploid. Females have an oogonium, males have a spermatogonium. Okay, so that's a diploid cell. It is going to undergo mitosis, and it is going to produce a B cell and an A cell. The A cell stays, and this becomes a stem cell that could then undergo mitosis again. So if males are going to have to produce millions, if not billions, of sperm during their lifetime, these diploid cells that were established in the testes cannot just develop into sperm, or you would run out of them, right? So they have to separate into A cells and into B cells, okay? And so the B cell is the one that is going to then go through meiosis 1, and it is going to be a primary spermatocyte, okay, so that's meiosis 1. And then it becomes a secondary spermatocyte. Okay, and then it becomes a spermatid. So this would be meiosis 2, sorry, I said meiosis 1 and I put meiosis 2. So this is meiosis 1, this is meiosis 2. Okay. So this is occurring as you start from the basal, laminar basal part of the seminiferous tubule, and then moving towards the lumen, which is the open space in the middle of the seminiferous tubule. Okay. So the spermatids are going to mature, and they don't actually mature until puberty, right? And then they start maturing, and then they mature at a high rate. So I'm going to just draw my little sperm. And one of the interesting things about the sperm is that they lose a lot of their cytoplasm. So they actually shed their cytoplasm so that they become very small. And so this is the nucleus of the sperm. This is the midpiece, which contains mitochondria. And they're a special type of mitochondria. They're kind of spiral in shape. And they are not going to contribute the mitochondria to the egg. So all the mitochondria that you inherit from the egg rather than the sperm. And then this is a flagellum. And so this is cytoskeleton element. So it's made of protein, right? And the mitochondria uses the fructose that's produced in the semen to power the movement of the flagellum, okay? We also have something really special here at the head, and it is a bag of enzymes. So I'm going to draw it like right there. And this is called the acrosome. And it contains enzymes. And this is what is going to help the sperm digest its way through the corona radiata and the zona pellucida. So it's got to digest its way so that it can eventually get to the egg, so that the nucleus of the sperm and the nucleus of the egg could fuse to form the fertilized egg, which is the zygote. Okay. So if we look at this in more detail, okay. So this shows the type A cells remaining in the basal lamina. This is the type B cell that is then going to undergo meiosis 1 and then meiosis 2, right, to form these early spermatids. And then notice that they actually shed their cytoplasm, and then they become spermatozoa. So the spermatozoa are the mature sperm that are then going to be stored in the epididymis and then ejaculated and then used to fertilize the egg. Okay. Okay. So if we look at this in more detail, So here's the acrosome, here's the nucleus. This is the special mitochondria. Notice that the mitochondria kind of migrate out to there, and then it gets rid of the excess cytoplasm. And so this is the whole structure of the sperm. And we saw that they were super tiny. Kind of interesting because in some species, they're not so tiny. So like some fruit flies produce giant sperm, but they produce very few of them. And the giant sperm actually, instead of fighting and competing for access to the egg, they kind of just like take up space. And so they're more likely, the first one in is more likely to fertilize the egg. But in humans, males produce a lot of sperm. And it's believed to be actually because there is sperm competition occurring in our evolutionary past. Females may have mated with a lot of males during ovulation, and then it took a lot of sperm, and the sperm actually, if you put two different males' sperm in a petri dish, they will actually attack one another. And so it's believed the reason why humans produce so much sperm is it's believed that we were once upon a time more promiscuous, more like maybe chimpanzees, where the female mates with the dominant male first, and then she subsequently mates with a lot of the subordinate males too. Okay.
Is the myth true about how they say like boys swim faster but girls live longer, as far as like between female and male sperm?
Okay, so that is really an interesting thing. The boy does do faster. There's actually more male embryos produced than female embryos, but male embryos are more likely to be defective. And so they have a higher, so like for every 100, they estimate for every 100 female embryos, there's actually 150 male embryos produced, but they're spontaneously aborted at a higher rate. Mm-hmm. And so that by the time they're born, there's generally like 105 males being born to every 100 females. But even male babies sometimes are less likely to thrive. And then males tend to die, have a higher death rate during like childhood and puberty than females. And they're driving fast cars. Yeah.
Jumping off.
Then suicide too. But anyway, and then
yeah,
I think males have a higher suicide rate.
I would assume males because I'd assume women because we get more emotional.
Yeah, maybe. Well, there's a difference between being males. If they try to commit suicide, they usually do. Females might try to commit suicide, but they don't do as well at it. It's a failed suicide attempt for females. Yeah. Okay. So we're going to compare this to oogenesis. And so this is actually, they might have rewritten your textbook. I forgot to look. But there's some new information about oogenesis. It was believed that at birth, all the oocytes are suspended in prophase 1 of meiosis. Prophase 1 of meiosis 1. Right? And so they think that there's like maybe 400, and these are what are called the primordial follicles. So when you look at the ovary, those are the ones that are kind of on the outside edge, and they're really little, and they have not started to develop yet. On the very outside edge, yeah, those are the primordial follicles. Now, there is new lines of evidence that suggest that there are oogonium in the ovary, and so there are stem cells, diploid stem cells in the ovary that are capable of undergoing meiosis, and so they're not sure what role those play in terms of like, do they ever do that? But there are stem cells in the ovaries, which in the past we didn't think there was, or they didn't think there was. Okay. So these are the primordial follicles. And then every month after puberty, these start to develop, right? And they develop into primary follicles. Right? So this would be meiosis 1. And then they develop into secondary follicles. And as they develop, the follicles become larger. And remember that they get thicker. So you have many layers of granulosa cells that are then going to produce estrogen, right? So they're going to produce the estrogen that is then going to cause the uterus to start to undergo mitosis, okay? So this is the secondary follicle. And then a mature follicle, so I'll put mature follicle here. So I'll put here it starts meiosis 2, but does not finish. Okay. So the mature follicle has a secondary oocyte in it. So this is the egg cell. And then it has a layer of granulosa cells. And then it has what is referred to as an antrum. And the antrum is a fluid-filled space. Okay. So this is my antrum. So that's my fluid-filled space. This is my secondary oocyte. Okay. And the secondary oocyte is going to be ovulated, and it is not going to be, it's actually going to be arrested in meiosis 2. And so it does not actually finish meiosis 2 until after it becomes fertilized with the sperm. So ovulation occurs, okay, and then you get fertilization. Okay, and then meiosis 2 is completed. Okay. So the important thing here also is that we do not produce 4 oocytes for every primordial follicle going into it. So we're just going to produce 1. So the question is, what happens to that extra genetic material? If we're just starting out with 1 cell that starts out in meiosis, how come we don't end up with 4 egg cells? And the reason for this is that we have what is called unequal cytokinesis. So unequal cytokinesis only takes place during oogenesis, does not occur during spermatogenesis. And so we start out with this primordial follicle, and when it finishes meiosis 1, it's going to have an oocyte, and then it's going to have a nucleus. Okay? And then this one can divide again. And then all of the cytoplasm goes with 1 nucleus, and then 1 nucleus gets left behind here. Okay? Does anybody remember what those nuclei that get left behind are called? Anybody know? These are called polar bodies. And it's believed that the reason why this occurs is that the egg needs to be large because it can only survive so long without being fertilized. And then it has to implant within a week in order to survive, because if it doesn't implant, it's going to run out of nutrients, right? And it's not going to be able to live. And so that cytoplasm, think of that cytoplasm as the initial nutrients that's needed to undergo cell division so that we can get this embryo that can then implant. And we'll talk about what that embryo looks like in a minute. So those are the polar bodies. The reason why they're called the polar bodies is that when the oocyte is ovulated, the polar body is actually right next to it. So it kind of looks more like this. And so remember that the oocyte is surrounded by a fertilization. It's actually a membrane. And this membrane is called the zona pellucidum. So this is the zona pellucidum. And this is produced by the follicle cells. And it is made of carbohydrates. So it's kind of sugar. And so it's like a membrane. And it's going to hold it there. And when we watch the videos, this would have been what they called the egg case, right? Or the egg shell. So this early embryonic development is going to take place in the zona pellucidum, and then the blastocyst is going to have to hatch out of that before it can actually implant into the uterus. So this is my polar body right here, okay? And then that would be the nucleus. So sometimes they use what is called a polar body biopsy. So let's say that you know that you are a carrier for a genetic disease. So let's say that you know that you're a carrier for cystic fibrosis. Maybe you even had a child that had cystic fibrosis already. And you wanted to make sure that your next child did not have cystic fibrosis. So you could have the doctor, the fertility doctor, would give you drugs to cause you to ovulate a bunch of eggs. Then they would take those eggs and they would remove the polar body. So they could just use a little syringe and they take the polar body out and they look to see if the defect is there. And so if the defect is in the polar body, then it's not going to be in the normal cell, right? So you can tell whether or not the oocyte is going to have the normal gene or the defective gene by looking at the gene that is in the polar body. And that's a pretty common way that they go about screening oocytes for genetic diseases. And it doesn't, you don't do anything to the oocyte, so you're just looking at the polar body and it doesn't cause any harm.
Do you have to put the polar body back in there?
No, you just take it out.
And if one of them does it, then they're all gonna.
So if it doesn't have it, then that means that the gene, no, if it has it, then that means that this gene would be normal. Yeah. Yeah.
But so if they're ovulating the egg and the egg and the polar body come out and you say, oh, this doesn't have cystic fibrosis or whatever, you put the egg back in and
You probably at that point, you would fertilize it with the male sperm and then you get an embryo and then you put the embryo back in there. So you would do it in a test tube. You would do the fertilization.
Is that how, like, designer babies are made though?
You could say that this is a designer baby, but really what they're doing is they're not actually screening for good traits. They're actually screening for a really bad genetic defect that they don't want to have in their child. Yeah. Okay. So here shows the primordial follicle, right? And then it's arrested, and then it undergoes, meiosis, And then here you can see the polar body, right? So the polar body would be right here. And this is after, or right before ovulation, right? So this is your mature, they call it the vesicular or the Graafian follicle, the mature follicle. And then it is ovulated, and then meiosis 2 doesn't occur again, doesn't complete until after fertilization. So the sperm comes in, and then meiosis. And here they sometimes show that the polar bodies also divide, but this is not necessarily the case. So here's showing this first polar body dividing into 2. Here you can see that right around the egg is the zona pellucidum. And then these cells, these follicular cells, are called the corona radiata, and they surround the egg. And then the follicle cells that remain in the ovary become the corpus luteum. So unequal cytokinesis occurs in the in oogenesis. Okay, I think that's something else I wanted to. We talked about some genetic screening. Oh, let's talk about genetic screening here. Okay. So this would be genetic screening by a doctor, right, of the embryo or fetus. So after 9 weeks, they generally call the embryo a fetus. So if this is done at like 16 weeks, then it would be the fetus. If it's done really early on, then it would be the embryo. So one most common way to do this is what is called the amniocentesis. Right? And this is where you use a syringe to obtain amniotic fluid. because the embryo is floating around in the amniotic fluid, it is going to shed some of its cells into the amniotic fluid. So then, once you have the amniotic fluid, you need to find the fetal cells, and then you need to analyze them. And one of the main ways that they analyze them is they do what is called a karyotype analysis. So a karyotype analysis is looking at the chromosomes. And so a geneticist is able to pair up the chromosomes so that they're about the same size and the same shape and the same banding pattern. And they can do it really quick, pairing them up to make sure that there is the normal number of chromosomes. And this is primarily done, as I mentioned before, for people that have Down syndrome, possibility of having Down syndrome babies. But it could be also or it could be done on a person that is an adult. You can also do a karyotype analysis if you think that something else might be wrong, like they might have an extra sex chromosome, for example. So another way to do this amniocentesis is not to use the syringe, but is to do what is called chorionic villi sampling. So this can be done earlier. I think it's like maybe 8 weeks. And what they do is they come up through the vagina. So they go up through the vagina of the female mother. Right. And they take a sample of the fetal part of the placenta. So they sample the fetal portion of the placenta. So if we look at a diagram that compares these two techniques, these two procedures, right? So this is showing it at relatively early. So this is the 8th to the 10th week, right? So this would be, at 9 weeks, it would be considered the fetus. And so they would just go up, and they don't actually have to worry about damaging or coming in and maybe poking the baby like they do here when they do the amniocentesis. But then this does carry a little bit of risk for causing a miscarriage if they go in and they mess with the placenta, but it's pretty rare. And so then this would be taking the fluid and getting the fetal cells, and then they obtain those, and then they look for the chromosomes and the matching up of the chromosomes. So if we look at the old way, the old technique, it was all black and white. But now they're actually able to stain the chromosomes. Do you guys have a question?
Well, I'm saying if they're testing for that, then are you probably like, that's kind of, I mean, I guess that's not designer baby, but like, what is that to like abort your baby probably if they have Down syndrome? That's kind of, or is
yes, so you can have that option, but if they don't do it in, it's kind of controversial because they won't do it in a Catholic hospital. So like if you needed to have, for some reason, if there was a bad genetic abnormality and you needed to have the baby aborted, then for us, we probably have to go to OHSU.
What other tests besides Down syndrome?
There's other chromosomes. There's 13 is a common one, and 18 is a common one. And then you can also have chromosomes starting to break. So cri de chat, the cry of the cat, is a broken chromosome number 5. And then you can also have some sex aneuploids, so you could have like only one X chromosome and no other, and then you could have XXY, which is Klinefelter's. So yes, so you could potentially give the mother the option of spontaneously aborting the baby or having an abortion in order to have a child with this genetic disease.
So 2 questions. One, if you found out I did have Down syndrome, is there like drugs that you can take for that? Is there giving you drugs? Like obviously you can't help it, but
To cure the baby or
Not
to
really.
have abortion?
no, like not to cure, but to like help it aid because they birth. Is that done?
So yeah, so it is due to a genetic abnormality, so there's no way that they could repair it. Yeah, there's nothing that they could do about
And
it.
then, is this something that you like opt for, or is this like, do they just automatically do it?
I believe you have the option. So you would get with your obstetrician and they would recommend that you have this done.
Because most of that they can detect through an ultrasound, like not like, oh, your baby has Down syndrome, but start to see symptoms and then give you the option for
Right.
the test, right?
So they can see problems with, well, obviously the heartbeat is important because if they can't find a heartbeat on a fetus, then it might stay in the mother for a long time without a heartbeat. And so then they give you the option of going in and having dilation and cutting the embryo out. They can also see whether there's deformities in the spinal cord, so they can see if it doesn't have a brain or if it has abnormalities in the spinal cord. And so then you have the option. Yeah.
Because then they won't just, I mean, if you kind of know what you're talking about, you go and you're like, I'm pregnant, I want these tests. Obviously, they'll give them to you, but if there's no reason for them to, like if they don't see anything on the ultrasound, they're probably not gonna.
When you get older, they really push amniocentesis because they want you to know if the baby's going to come out with Down syndrome. They don't want it to be a surprise.
Yeah, because that's pretty easy to detect that within like the first, what is it, 5 to 16 weeks or something
Right.
like that.
Yeah. Okay. Okay, so we're going to start on the very last chapter of the book today. And this is pregnancy and gestation. You're done.
After this chapter.
I still got 2/3 to go.
Just wait till you go to, I have people come back from nursing school and they're like, I'm really sorry I gave you a hard time for everything we had to memorize because nursing is so much more memorization. And I can't believe I gave you such a hard time.
Oh, I'm not giving you a hard time. I'm just like excited that.
I can't believe that.
Oh,
I thought A&P was hard.
A&P is hard, but nursing school is 10 times harder. But I'm not complaining about it. I'm just so happy 3 terms of this class and we're done.
I think the difficulty depends on where you go to, though.
Yeah.
Pharmacology doesn't even make any sense. They can just name drugs whatever they want.
Yeah, pretty much. They said if you ever want to get rich, just make a drug. And then patent it, and you'll never have to work at it another day in your life.
You have lots of legal insurance.
Well, I don't mean like get high drug. I mean like a pharmaceutical drug.
And those drugs, that's kind of scary. Okay, so about 128 days.
Oh, that's a lot of days.
But interesting, our gestation is not counted at the time of fertilization.
280 or what? Yeah.
You said one and then you
280 days.
You just said 180.
Oh, I meant 280. Okay. 280 days. So it's not counted from fertilization because we can't see fertilization. In humans, ovulation is hidden. If you think about other animals, even other primates, when a female is ovulating, it is obvious. Like 1, she signals it, and then 2, like her external reproductive structures get engorged and get bright red, and it's just like obvious, right? Cats, you know when they're ovulating because they go into heat, right? Humans, we hide ovulation. So we do not know when we're ovulating. You can now get tests for ovulation over at the grocery store, right, to find out when you're going to ovulate. Interestingly, our body temperature changes. I think it gets warmer right before we ovulate. So sometimes women try to figure out when they're ovulating. But so what is obvious in human reproduction is menstruation. So it is counted from the first day of the last menstrual period, right? So counted from first day of last menstrual cycle. So that's why they want you to keep track of this, because they actually use that to determine when the baby is going to be born. Right? They estimate the birth date based upon that. Okay? So what that means is that upon fertilization, you are technically 2 weeks pregnant. Right? So at fertilization, you are 14 days pregnant. So
your due date is not like so scary, I guess, if you have a 2 weeks leave? Or do they just advance you?
they count that in 280 days. So at fertilization, female is technically, boy, this pen is just, I need a new pen. Technically 2 weeks pregnant. Okay. So in order to understand how fertilization needs to happen when coitus, when copulation needs to occur, when the sperm has to be present, when the oocyte is present, we have to look at the life cycle of the egg. So the oocyte can only survive 12 to 24 hours after ovulation. That's after. Okay. The sperm, on the other hand, have a longer life cycle, so they can survive 24 to 48 hours after ejaculation. So in order for fertilization to take place, or to prevent it from taking place, you could say that ejaculation must occur no earlier than 2 days before ovulation or 24 hours after ovulation, right? So we could say coitus. So that would be the male and the female coming together, the sperm being ejaculated into the female's vagina, right? Must occur no earlier than 2 days before ovulation or 24 hours after. Right. So you have this window here of when the egg is fertilizable, right? When you can have a baby. And this is, sometimes people pay attention to this to prevent pregnancy, right? So the rhythm method, which doesn't tend to work very well because ovulation can occur at different times. But the rhythm method would be paying attention to this so that you do not have sex during the fertile period, right? But also it's obviously important if you're trying to have sex that you know when it's best time to have sex in order to get fertilization to occur. Okay. So if we look at the embryo and the fetus, okay, so at fertilization, we have, and up until 5 weeks, we have the embryo, no, 8 weeks, I guess, 8 weeks, we have the embryo. And then after 9 weeks, we would refer to it, the developing person as a fetus, okay? So 1 to 8 weeks, it's an embryo. 9+ weeks, it's the fetus. Okay. So let's talk about the things that have to happen in order for sperm to fertilize the egg. So remember that the vagina is not necessarily a very hospitable environment for the sperm. What does it have that kills off the sperm readily? Acid, right? So it has acid because it is feeding bacteria that are producing lactic acid, lactobacilli bacteria, and so the vagina is very acidic. So we have to have lots of sperm, right? So we have to have lots of sperm. I think there's, oh gosh, I can't remember what the cutoff, millions of sperm, 4 to 5 million in every ejaculate. See if I can. think I forgot to write that down. Anyway, lots of sperm. The other thing is that the sperm actually must sense the egg so that they know how to swim and the direction to the swim. So the sperm are said to smell the egg. Okay. So for some, it's believed that they have receptors on them that can detect chemicals that are produced by the egg, and so they move towards those chemicals. So this is what is called chemotaxis. So they swim towards the eggs, toward the egg based upon the chemicals that the egg is releasing. The other thing that the sperm must do is they undergo, so the sperm undergo capacitation. And we saw this in the video, and this is where the sperm rub themselves against the lining of the fallopian tube. And this is actually believed to strip off some of the proteins that are on the outside of the sperm head region, so that when the sperm does get to the egg, it can actually release its enzymes. So this makes the sperm able to release their enzymes. It makes them able to release enzymes. Okay. Then we have what?
Before you continue, is it true that like there's a pre-ejaculation that has more sperm than like the actual ejaculation?
No, actually it's the reverse. Okay. So we talked about the bulbourethral gland, and it produces pre-seminal fluid. And that pre-seminal fluid could have sperm in it if the male has ejaculated recently, but generally it doesn't. So the pre-seminal fluid doesn't generally have a lot of sperm in it. It is the ejaculate that has the sperm in it.
Anybody else hear that in like high school health?
Yeah,
I just
I
heard
know
it.
what you're talking about. Yeah, like they always said we found being pregnant.
Yeah.
Yeah, they're just warning you to take precautions. Pre-seminal fluid. The pre-seminal fluid. That's the bulbourethral gland, right? We talked about how that lubricates the urethra and gets rid of the urine, right? And it could have some sperm in it, but it doesn't generally, unless the male has ejaculated recently and then hasn't urinated, right?
Which is why when you're 16, you'd be afraid of it.
Yeah.
Because boys at 16.
Okay, so then we have what is called the acrosomal reaction. So this is necessary because you remember that the oocyte is surrounded by layers of cells called the corona radiata, and it also has the zona pellucida. So this is an enzyme reaction that starts to break down that barrier. And actually the first sperm that gets to the egg is not the one that gets the egg, because you have to actually have hundreds of sperm undergoing this acrosomal reaction in order to kind of break down the wall in order for one sperm to get in, right? So this must be accomplished, or must be done, by hundreds of sperm just 100 plus sperm before one sperm can break through. And fertilize the egg.
Now, what stops multiple?
We're going to get to that in a second.
What do they break through?
They break through when we look at the oocyte. I'll draw it here. Here's my oocyte, and it's been ovulated, and it has this zona pellucida around it. And then it also has these cells which are called the corona radiata. These are the cells surrounding it. So this is the radiating crown, corona radiata. And then this is my zona pellucidum. Okay. So in that last chapter of your book, actually it's not the last chapter, it's chapter 28, isn't it? Chapter 29 is heredity. So chapter 28, there's some pictures that show this really nicely. So this is the zona pellucidum right here, right? These are the granulosa cells, so that would be the radiating crown. So that's the corona radiata. This right here is demonstrating the acrosomal reaction. And so here the sperm releases its enzymes and digests its way. And here you can see that this one sperm has actually broken through, and this is really important. There's actually receptors on the outside of the egg that detect the sperm. And this actually means that the sperm has to be of the right kind, right? So if you think about other creatures, if you think about sea creatures that just release their sperm and eggs into the water, there's lots of sperm and egg of different species floating around together, right? So how do you know what egg and sperm can come together? And it is because they have the proper receptor and the proper binding protein. And so this sperm here is going to bind to this receptor. So this is called sperm binding receptors, right? So the sperm, next thing, must bind to receptors on the outside of the egg. So this is actually one that is broken through, I guess, right here. And so we have mechanisms that prevent, hopefully prevent, more than one sperm fertilizing the egg. Okay? And so that is what is referred to as blocks to polyspermy. Okay? So what blocks the entry of more than one sperm? So that's called polyspermy. So the first thing is that we have a fast block. And from the video, you saw that as soon as the sperm entered the egg, this electrical wave moved across the surface of the egg. And so that is depolarization of the egg membrane. And that's due to an influx of sodium ions. Influx of sodium ions. So sodium floods in and it changes the electrical charge of the membrane. And so that prevents any other sperm from coming in. And then we have what is called the slow block. The slow block is due to a release of calcium ions and this causes, I'll show you a picture of this in a second, which causes cortical granules. That's a T, cortical, I think there's an A there, cortical granules to be released. And this creates the fertilization membrane. So the slow block has another name, and it is called the cortical reaction. So if we look at this part of the blocked polyspermy, oh, maybe it was on the previous one. Here it is. Okay, so calcium starts to accumulate inside the oocyte. So calcium is released into the cytoplasm of the oocyte, and then it causes these little vesicles, which contain cortical granules, to fuse to the outside membrane. And then the cortical granules exit that membrane, and then that produces a hardening of the zona pellucidum, which leads to that fertilization membrane. So I'll just put hardened, let's see, cortical reaction, fertilization membrane. And so this is the hardened zona pellucidum.
Is the function of this fertilization membrane just to like protect it? Or?
Well, it keeps any other sperm from getting in, right? And then the early embryo, it keeps it together. So if for some reason it doesn't, then you can get the embryo splitting into two, and then you could get identical twins being formed. But it kind of keeps those early cells from splitting up and keeps them together, and it keeps, it's a slow block to polyspermy. Okay, so I'm going to stop there for today, and then we'll finish up on pregnancy on Wednesday. And make sure if you have any questions that you come and ask them about the review sheets. Excellent, thank you. And I have your quiz. Okay, actually, why don't you come downstairs? That'd be better for me.
I think you can take the wrong one online. Because I have one of them in and then one of them was still 0 out of 0. But I did both of them.
You know, do you realize that it takes some days for it to...
I think mine is still 0 out of 0.
Well, it came up, like both of them, both of my grades came through, and then when you deleted it,
okay.
it went missing. So that's why I'm thinking.
Okay, yeah. I'll check. I'm not sure what's going on there. I'm confused. I can't quite figure out which one is which. It won't give me that information.
Yeah, it's kind of...
I should just keep them both up there, and then whichever one you do,